Hi, my name's Reece, and today I will be presenting questions 1 and 2. So, which possible samples would you take to send to the laboratory? According to the Lumpy Skin Disease Field Manual compiled by the Food and Agriculture Organization of the United Nations, samples to be taken are of lesions and scabs from the dermis, saliva, swabs of the inside of the animal's nostril, blood for the polymerase chain reaction test, as well as the ethylene diamine tetraacetic acid test, as well as whole blood for serum. Now, how to preserve and transport these samples. Samples should preferably be obtained from live animals before antimicrobial treatment. If the animal is deceased, samples should be collected before the onset of autolytic or putrefactive processes in the corpse. The refrigeration of specimens should be required in the occurrence of warm and humid weather. As LSD is a viral disease, it can be frozen to ensure the integrity and preservation of the specimen and contain pathogens. Specimens should have their own container that completely isolates it from the elements and outside contamination. These containers need to be labeled with the specific animal's details, such as identity, sample type, and the date it was collected. Specific transport regulation of the country or area must be adhered to, such as secondary and tertiary packaging. The sampling method must ensure not to cause conditions or changes that will hinder the accuracy of the test to be taken later on.
Hi, this is Calah Pillay. Tests that can be done to check if this is a viral infection include an antibody test, a viral antigen detection test, a viral culture, or a viral DNA or RNA detection test.
It's MJ Mentor. My question was, how will you be able to confirm the morphology of the virus, and which morphology can this virus possibly have? Confirmation will be through viral isolation. And like other viruses in the poxviridae family, capillary pox viruses are enveloped with brick-shaped geometry. They have a linear genomic arrangement and are monopartile with average size of 220 nanometers by 260 nanometers.
Hi, I'm Almarie Kolbe and I'll be doing questions 5 and 6. The most likely virus to cause skin lesions in cattle is of the family Poxviridae and of the genus Capillary pox virus. It is more commonly referred to as lumpy skin disease or LSD. The Capillary pox virus grows to a high titer in a wide variety of cultures, according to an article on lumpy skin disease by Professor J.A.W. Kutzer and Dr. Eva Tuparainen. It grows very slowly though and may require several passages. The primary cell used in cell cultures to grow the virus is Lyme testis cells from exotic merino sheep. However, this source of cells is getting too expensive to obtain since less farmers breed merino sheep. As an alternative source, fetal bovine skin cells and fetal bovine muscle cells can be used. Exotic breeds of sheep and cattle are used because the virus yield is 3 times greater than that from local breeds.
Hello, I'm Jacome Pretorius and I will be answering the following 2 questions. Question 7. Some fungi cause skin lesions in cattle. How will you determine this is not a fungus causing these lesions or another virus? To differentiate whether it is a virus or a fungus causing the lesions, a UV light can be shone onto the affected area. If it is a fungal infection, the affected area will glow under the UV light. A viral infection will not glow under the UV light. The clinical signs and lesions of lumpy skin disease can be confused with infections from another form of virus, for example, as seen in pseudo-lumpy skin disease, which is caused by bovine herpes virus 2. However, pseudo-lumpy skin disease causes more superficial lesions, while lumpy skin disease has much deeper lesions. To confirm the diagnosis, specimens can be taken from the affected area and sent to a laboratory where it can be studied under transmission electron microscope using negative staining. Question 8. Explain the specific eclipse phase of the replication of this virus. The eclipse phase is the phase during which the virion has entered the cell and before progeny viruses are made, during which the virus gains control of the host's synthetic machinery and produces components required to assemble the progeny viruses. The Capillary Capripox virus contains double-stranded DNA, and its replication is cytoplasmic. It enters the host cell by attaching its viral proteins to the host cell's glycosaminoglycans, which mediate endocytosis of the virus into the host cell. It fuses with the host cell's plasma membrane and releases its genetic material into the host cytoplasm. The host cell's enzymes transcribe the viral DNA in the nucleus, and the viral enzyme is used to transcribe the viral DNA in the virion.
My name is Kirthi Singh. The question given is this virus prone to mutations, and what are the possible types of mutations that can occur in this virus? Pox viridae is a DNA virus that consists of DNA as its genetic material. It is therefore not prone to mutations. DNA viruses copy their genetic material by utilizing enzymes from the host cell called DNA polymerase. These enzymes proofread the strand and correct the mistakes as they go. RNA viruses have a high mutation rate due to the fact that they use enzymes called RNA polymerase, which don't proofread and therefore makes many more mutations. The possible types of mutations that can occur are, number one, antigenic drift, is where individual bases in DNA or RNA mutate to form other bases, which result in minor changes in antigens. These are mainly point mutations which have no major effects. The second is antigenic shift. These are mutations where there is a major change in the genome of the virus, process by which two or more viruses, which can be a strain of that virus or a different virus, combine to form a new subtype consisting of a mixture of the viruses. This results through recombination or reassortment.
Hello, I am Nadine Daniell, and I will be discussing the transmission of the lumpy skin disease virus as well as its shedding into the environment. There is no definitive mode of transmission for the lumpy skin disease virus. Direct transmission by contact between animals has been proven to be inefficient. Biting insects like mosquitoes and ticks has been proven to play a part in infection. The yellow fever mosquito transmits the virus mechanically, as well as the brown and bond tick males, whereas the African blue tick transmits the disease via transovariant transmission, which means the parent tick transmits the virus to its offspring. The virus is also spread through common drinking troughs, which is infected with infected saliva. Infected cows can give birth to calves with pox lesions in the skin through vertical disease transfer, and suckling calves that drink from a mother that is infected can get infected through the milk. It's also been proved that the virus is persistent in the male genital tract, which means that seminal transmission is possible. It is unknown if it's through natural mating or artificial insemination. The virus is spread into the environment when the animal starts to secrete saliva as well as ocular and nasal discharge. Bond ticks and African blue ticks have mechanisms of overwintering, which may explain how lumpy skin disease overwinters during inter-epidemic periods. The virus is also remains viable in a scab or tissue fragments for several months, meaning that any scab or infected tissue that is spread into the environment can infect other cattle when it comes into contact with them.
My name is Kirthi Singh, and the question given is, being an intracellular pathogen, which mechanisms can this virus use to evade the immune system of the host? Intracellular pathogens are capable of growing and reproducing inside the cells of the host. Mechanisms that they can use to evade the immune system are, number one, non-cytocidal infections. These are viruses that don't kill the cells in which they replicate, but rather cause a persistent infection. The infected cell produces and releases virions while it continues to grow and divide. Number two, induction of immune tolerance. This is a state of immune unresponsiveness that is specific to a particular antigen induced by previous exposure to that antigen. Number three, Immunologically privileged sites. These are anatomical sites within the animal body which provide an environment that favors a prolonged survival of antigens or viruses. Number 4, integration into the host cell genome. Integrated viral genome can remain latent and passively replicate along with the host genome. The virus can be reactivated, leading to viral transcription and production of new infectious viruses.